Okay. Here we go. Starting. So, hello everybody. This is your Chapter 11 lecture video for this. We'll start it over again. Okay, and go. Hey everybody, this is your Chapter 11 lecture video. We're going to get into some DNA technology specifics here. So kind of building off of chapter 10, where we started to look at just different aspects of DNA and how we use DNA. Now we're going to kind of go into some stuff that you've actually seen in lab. We've done PCR and gel electrophoresis. So you're going to see a little bit of that stuff kind of coming back into here. So first of all, what are some of the tools of DNA technology? Why do we have DNA technology? So ever since we've been able to figure out the structure of DNA, that double helix model, we've understood now that it's the inheritance model. It is the molecule that's carrying our genes along, that's passing our genes along, allowing us to continue to pass along our traits. Once we kind of figured that out, we then started to see, well, how can we start to predict and maybe even change that? Please make sure you take some time to read at the end of the chapter and at the end of the soft chalk the ethical concerns around DNA technology and manipulating DNA. There are a lot of kind of controversies that can come up with it. There are a lot of conflicting opinions that are not science. So that's where it gets really tricky, because the science is one thing, but the science doesn't tell us if something is good or bad. The science can't tell us if something is helpful or harmful, really, in the long-term sense. It's hard to predict how a change in a DNA molecule at one level will be inherited along, depending on what kind of traits you're manipulating. There are some controversies that come up with DNA technology, and depending on where you are in the world, different governments have different regulatory practices for it. But the scientific community at a large tries to regulate itself as best as we can. So just kind of keep that in mind as you go through this. So again, looking at some of the tools of DNA technology, things like recombinant DNA and DNA sequencing. So recombinant, you might have remembered recombinant chromatids, sister chromatids during crossing over. So it's when you're putting different DNA into another molecule. PCR we've seen, and DNA profiling. We've done a little bit of that in our classes as well. We can look at stem cell research and cloning. You probably have heard of some controversies. You probably heard of that in the news. It usually kind of comes up every couple of years. Some change or some breakthrough happens, or some old controversy gets brought back up again that we kind of have to rehash out. DNA and technology, this is another big one. DNA and pre-implantation genetic diagnosing for IVF, things like that, just normal pregnancy as well. Genetic testing, gene therapies, those kind of things. So we're going to look at all that. It's important to remember that DNA is the molecule of protein synthesis. It's the basis of protein synthesis. And all organisms on Earth share that same common language, if you will. All organisms on Earth are made of DNA. We all use proteins. We use the same DNA molecule. We use the same amino acids. And so as long as you are providing a cell with those requisite materials for protein synthesis, and you should be able to remember what some of those things are, things like a DNA template, right? Your polymerase enzymes, your RNA polymerase, the ability to cleave and create exons, so modify your mRNA, amino acids and tRNA, ribosomes, those kind of things, right? If you remember, all cells pretty much have those things. And so if you have that, if you give that into a cell, they'll be able to make proteins. It's one of those, you can do it in a test tube. I keep saying a cell, but if you can do it, you can do it in vitro. Or in vivo, vitro. You can just, in a test tube, mix all the requirements that you need for protein synthesis, and it'll start to make proteins, as long as all the chemistry levels are correct in that test tube. And so with transgenic organisms, we're able to take DNA from one organism and put it into another. And that new organism can then express those genes because, again, we all use the same DNA molecules and we all use the same things for protein synthesis. So here you're seeing these glowfish. These were really popular when I was in middle school. We had several of them because they tend to not live very long. But they have been modified with a gene from a sea anemone that allows them to glow. And so that's just a protein. And so they just kind of put that protein and that gene into these organisms' genome. They add that trait into there, and then now those proteins are synthesized and these fish can then glow. The fish is a transgenic organism. The genome, the DNA inside of that fish's cell is called recombinant DNA. It's been combined with other DNA, foreign DNA, to make something new. So it's now a transgenic organism with recombinant DNA. So it depends on how you want to study that. You might study the organism itself after the implantation, after the DNA has been put in there, or you might be studying the process of putting DNA into something. So two different kind of aspects there. So how do they do that? How do you create a transgenic organism? This was the slide before was about a fish, but this is true for plants, for animals, for all kinds of things. This is not me telling you to go out and do this, okay? You need some very specific tools for being able to do this. But it is essentially a pretty kind of straightforward process when you're working with simple things. Once you start getting more complex organisms, it starts to get a lot more difficult. So the basic way of creating a transgenic organism is to find the DNA that you want to, take the host DNA, cut that section out, put your section in, and then hopefully it takes up. A lot of different factors can go into that process. For example, when you go to cut the host DNA, when you go to kind of sever the host DNA, it might just zip itself back together again if you're not able to get the foreign DNA put in there at a time. So a lot of times when you're creating recombinant DNA, you're doing a bunch of it. You've probably already done some PCR to make a bunch of copies of what you want to make, and then you put it all into a test tube. You kind of put it at the right conditions, and you hope that some of the DNA that comes out will be recombinant. Some of it won't. That's just the nature of it, because like I said, things will join back together. But maybe you get good. Maybe you start to see, oh, I could adjust the pH of the solution a little bit, or the salinity or the temperature. If I adjust these things just a little bit, that actually makes the DNA of the host less likely to join to itself. And that gives my DNA, my foreign DNA, more time to get in there and bind in there to the host DNA and insert itself in that. That's that process of inserting yourself into the DNA, or inserting recombinant foreign DNA into the host DNA. So there is a technique. It is a skill to be able to do this, and it has to be done at a large scale for it to really be successful. So example here is how we make insulin. Insulin is a hormone that helps to regulate blood sugar. It is absolutely vital. It is one of those things where it is incredibly expensive. The process, the buying insulin is much more expensive than it needs to be. The person who made insulin has kind of shared that process with the world. And so, you know, that's just kind of where we are. And so if you know anything about healthcare in America, you probably have heard about the prices of insulin. And here's how they do it. Here's how we can start to make insulin, human insulin. So what they do is they take the pancreatic cells, human pancreatic cells that make insulin, that's part of the process is they're synthesizing that hormone of insulin. They take the DNA from that cell and they snip out that section that's used to make insulin, that insulin coding section. Then they hold on to that, they kind of put that to the side and say, okay, thank you, that's our acquired DNA. That's the DNA we want to put in. Okay, so you can see that kind of following on the screen here. So that's the one we acquire our protein coding DNA, which in itself is a process because you have to figure out in an entire genome, and again the entire human genome, you have to figure out exactly where it is that codes for insulin. And we'll kind of talk about how scientists start to do that later when we get into our DNA sequencing. But once you have your gene sequence, you kind of put that to the side, and now you say, okay, now we need a cell that can make a bunch of this. Because with people that have diabetes, a lot of times they're not able to make their own insulin. And it can be really expensive to make insulin in a lab. But bacteria are essentially little factories. Bacteria are really good at just taking something and making a whole bunch of it. They replicate really fast, and so they're great little workers. So what scientists will do is they'll take bacterial DNA, what's called a plasmid. A plasmid is a circular DNA. It's a ring of DNA rather than our double helical DNA. So it's a circular ring of DNA, and they go in and they cut the plasmid at a specific spot. And the ends, they call them sticky ends. And the reason they call them sticky ends is because they're able to, because they know the sequence of insulin and they know what the nucleotides are at the end of insulin, they can essentially match those ends with the ends on the bacterial plasmid. They can get a restriction enzyme that will cut the bacterial plasmid at exactly the right point so that when it snips, these ends, you can see the nucleotide ends, those ends are going to match what's on the end of your insulin strand, which just essentially is kind of making sure it's kind of amplifying that effect of them joining back together, again, sticky ends. This idea that, okay, yes, the bacterial plasmid is going to want to join to itself, but if our insulin is also complementary to those nucleotides, we might be able to get our insulin to bind first. And so that's one of those reasons why you have to be really specific. You actually have to know your sequence before you can do any of this, your DNA sequence very specifically, before you can really do this. But once you do that, if, again, you're mixing your donor DNA with your host DNA, hopefully it takes up. And when it does, you create now recombinant plasmids, recombinant DNA. You put that back into a bacterial cell, and that bacterial cell, those ribosomes will just start churning and burning insulin out. You can then harvest that insulin, and then depending on what country you live in, sell it for a major profit. That's for plants. That's for bacterial cells. This is for non-mammal cells, non-animal cells. Animal cells are much more complex. They take a little bit. They do something different for them. But for plants, for something like this, right, you can kind of do it like that. Plants, we can use bacterial tumor-inducing cells, is what it's called, because you need something that's going to be able to insert itself into the host. Bacteria don't have the ability to insert their DNA into our cells, into mammal cells. Mammals have, or animals rather, not just mammals, but animals have more complex kind of mechanisms to be able to make sure that we don't get infected by bacteria. Viruses are really good for that. So we'll see that for animals, you have to use viruses to make recombinant DNA. But for plants, they don't quite have the same check and balance mechanisms that we do, the same immune protection that we do. And so you can use what are called tumor-inducing plasmids from bacteria to insert genes into plants. Plants were able, the tumor-inducing plasmid essentially is able to enter into the plant cell and then join with that DNA. You can add it into the host DNA. Will infect it basically. And so what you're seeing here is an unaltered tobacco cell, and this one is a transgenic tobacco cell. It's been added with an herbicide resistance gene. So essentially, this allows farmers, if you have a plant, a tobacco plant that is resistant to herbicides, you can spray your entire field with herbicides to kill off the weeds, the non-desired plants. I'm not going to call them weeds. I'm going to call them the non-desired plants without affecting your crop that you're trying to grow. And so it's just a way for being able to make sure that the plants that you want to maintain kind of have that protection against them. And so essentially it's just pretty much that. You kind of create your recombinant TI plasmid, just like TI is tumor inducing. You create your recombinant plasmid like you did on the slide before, and then you just put that bacteria onto your cells. You kind of take your little swab and you kind of inoculate. That's the word I was looking for. You inoculate your plant cells with these bacterial cells, and those bacteria will move in there. They'll insert their DNA, and the plant will start to produce these proteins that prevent it from being resistant to or prevent it from dying from herbicides. Like I said, with animals, you have to use viruses to create transgenic animals. Essentially the same thing if you think about how a virus works. Viruses are these little protein capsids that are going to get into our cells. They're incredibly, incredibly small. They get into our body, they kind of climb onto one of the cells, and they inject their DNA into our host cell. And then that DNA is essentially then their DNA kind of takes over all of our internal machinery, and they start replicating their own proteins. Those viral proteins then start to spread, and they make us sick, right? Or they, kill us if they're really bad. But what happens is with those, if you can make a virus that is not going to alert your immune system, and if you make a virus that is not going to cause some kind of major internal disruption, if it's just a virus that's just adding in this extra protein that makes you glow, right? That's not then causing a tumor to grow. Oh, it's just causing this protein that's going to be broken down and recycled. And so you're able to create kind of these recombinant animals. And as they divide, their cells are going to pass that along to those dividing cells. We're going to talk about if you don't modify the sperm and egg, those traits aren't going to be passed along. You have to modify the gametes, the actual sperm and egg, for this fish to pass along those transgenic traits, which they have. Right. Otherwise, you're having to modify every single fish that is born if you don't modify the gametes. So going back to DNA sequencing, this is how we can figure out what that insulin gene is, right? To be able to figure out where the actual insulin gene is, what are the nucleotides on the end, what are the nucleotides on the plasmid, how can we then create these sticky ends? This is all DNA sequencing that you have to do. And so on SoftChalk, there should be a video called the Sanger sequencing method, a video that kind of goes over that. I would recommend watching that just because this can be a little tricky to understand if you kind of don't see a visual representation of it happening. So go check that video out if you haven't gotten a chance to. But essentially with a DNA sequencing, you start with an unknown DNA sequence. So let's say that we are talking about trying to figure out insulin. Well, we know insulin can cause diabetes. Okay, so essentially what you start with are people with diabetes. Who are all the people that have this same condition? You get them all together and you take them, not all at once, obviously, but you take those people and you take a blood sample and you look at their genome. After the Human Genome Project, we kind of sequenced our entire genome. We were able to start looking at all these things. And so once you kind of have everybody's genes sequenced out, you can start to see, well, where do they have matching sequences? Where do they have matching maybe be errors or mutations in their DNA that all of these people with diabetes have. So you have a population that you know has something. These people have diabetes. Okay, now let's look at their DNA. Where are the matching points? Where does everything line up? Okay, well, that sequence where all the people with DNA, they're all with diabetes, they're all going to have that same matching gene. That's your insulin gene, or the one that's messed up. That's your insulin gene. That's the miscoded gene. Okay, so now we can go on someone that's healthy. We compare that to healthy DNA. We find that same location because genes are physical things on chromosomes. They're not just floating around. They have a physical location on a chromosome. So if you know where the wrong one is, you can look at a healthy person, figure out the right one, take that one, put it into there. That's kind of what it does. That's not what we do. Obviously, we take that correct sequence of the healthy insulin coding sequence. We put that into bacteria. The bacteria make the insulin, and then people inject insulin into them. We are not when someone is getting an insulin, when someone has an insulin pump or getting insulin injection, that is not gene therapy. I'm gonna have to make sure. Let's say that one more time. An insulin injection, an insulin pump is not gene therapy. We take the insulin gene that gets put into some system that makes the protein, that makes the hormone, and then you inject that into yourself. So it's not gene therapy when you're doing insulin injections. But it is a type of technology that we use that uses DNA to create that final product. And so with this, with when you're sequencing DNA, right, you take kind of your unknown sequence. Okay, this is the gene. We know this is the insulin gene. We don't know what the incorrect sequence is or the correct sequence is. But if we take all the requirements, all the things that you need for DNA synthesis, DNA replication, think about what those requirements are: your nucleotides, your polymerase enzymes, your primers. If you put all of those into a test tube together, you can start to create this gene, this sequence of genes. And what they do is it used to have to be one at a time. Now that they have computer models, it's more advanced. They can use modified nucleotides that are flagged with a radioactive kind of color. And so that lets them see. Well, as the gene, once they kind of put everything in together, they kind of put the unknown DNA with all the primers, everything in there together. What they start to notice, what starts to happen is that you get these kind of varying lengths, because these nucleotides, they're modified to both be radioactive and glow, but also to be terminators. So you can't add anything to them. And essentially what that starts to create is this sequence of different sizes. So let's say that for a sequence of genes, let's say the sequence is ATCGA. Well, as you go along, as you start to kind of figure out your DNA sequence, you're going to see increasing lengths of DNA. The shortest strand, that might be that A, because again, it's going to be a terminator. So all you'll see is just that one A nucleotide. The second strand is going to be a T. So you'll be a little bit longer and you'll see the T nucleotide. The third one is ATC. And so you'll see that C nucleotide. And so it just kind of keeps on going stepwise, longer and longer. And then you just read those bottom ones and that tells you what your DNA sequence is. Watch the video. There's the video link if you need to. It's on Softalk. It's helpful to see it kind of visualized in that sense. But essentially, you just kind of take the unknown sequence, you mix the nucleotides together, and it starts to come out. Now, that's the nice way, because someone else has already figured it out for us. What's complicated, though, is that most human DNA is non-coding. Only about 1.5%, a very, very small percentage of our DNA is actually used to make proteins. If you remember, introns are removed, exons are expressed. Well, even the non-modified mRNA is still just a small snippet of your entire DNA sequence. There's a lot of untranslated, redundant sequences within our DNA. And so you can kind of see this here. You have the exons of the purple, the pink are the untranslated region. And then as we go down, humans, chimps, we share a lot of the same kind of intron, exon related regions. You see with mice, there's fewer of those, still some, which is why we can use mice as model organisms for humans when we do like a testing in labs. Zebrafish, you can see there's really not a lot of overlap. There's some overlap within our exons, just coding for normal, proteins that animals have to have that make us all animals, that put us all within that same kingdom at least. But there's really not a whole lot of other genes that are being put in there, not a whole lot of other DNA regions that overlap with humans in terms of zebrafish, which is why we really can't use zebrafish as an organism for how humans would react versus like mice or rats or chimpanzees or orangutans. So this is again just that idea that there is a lot of DNA that is non-coding, and so when you're talking about creating the human genome and sequencing the human genome and trying to find one specific gene, there's a whole lot of mess you're just kind of working through before you actually find the correct coding sequences. So let's say that you do find that. You do find your insulin gene and you're trying to put that gene into your bacterial plasmid. Like I said, you need a lot of it. You can't just do one and one. You need millions of these. And so we use PCR. And we've seen PCR. We did it in our lab a couple of weeks ago. This is your polymerase chain reaction. Listen to the name of things. Polymerase. This is a polymerase enzyme. So you're going to be creating a lot of copies of one thing, long strands of copies of things. This is helpful because, you know, if you are at a crime scene investigation, I was saying the insulin one, a lot of times with this kind of DNA stuff in these next couple of slides, we'll be talking about like crime scene investigations, where if you have a little bit of DNA, you want to be able to amplify it so you can send it to different labs for analysis, send it to different departments for comparisons, those kind of things. Save it for, you know, future use. If the case is, you know, starts to go 10, 20 years old, you want to be able to have multiple copies of that DNA. So similar to what we were doing with our gene sequencing, once you kind of find the DNA, you isolate the DNA that you want, you can separate that and then start to make copies. You use, in this case, we use a primer. We use a specific primer that is going to bind to the beginning of the gene that we want, and it will just make sure it runs through that gene. Because if you remember, at the end of every gene, they have a terminator sequence as well. So our primer makes sure that we are just kind of copying that one sequence. If you're trying to copy somebody's entire genome, then, you know, you are using multiple primers. One of the ways that we can start to use this technology is through CRISPR. CRISPR, I was probably in high school or college when CRISPR was really like becoming used. You know, it was one of the things where we heard about it for a few years, and then you start to actually hear about it kind of being used industrially or kind of at a larger scale. So CRISPR is an acronym. I don't remember what it stands for. It's you know, it has a long name. It's kind of like laser or NASA or one of those things. Like it has some meaning, but I don't know what it is. SCUBA is another one of those. So CRISPR is essentially a system. You're using multiple different techniques to be able to find a gene sequence. Like you find the gene sequence, you can cut those nucleotides and then add a new gene into there or modify it somehow. So CRISPR was really innovative because it allowed us to kind of start modifying human DNA too, start actually going through gene therapy. That's kind of what it started to introduce for us. So with CRISPR, it uses an enzyme called the Cas9. CAS, the Cas9 enzyme. And so you start with guide RNA. So you have the sequence of RNA that's going to be complementary to your DNA. And that RNA kind of goes in and boom, it's going to bind to the DNA sequence that you want to modify. Once the RNA has bound to that DNA, then then the Cas9 enzyme is going to go and it's going to bind to the RNA. And when it does that, it's going to snip your DNA at a specific sequence. So you can see this DNA cut site where the Cas9 enzyme is going to go and it's going to snip at that RNA. So the big thing with CRISPR was making this Cas9 enzyme, this enzyme that was very specific. It wasn't just going to go into somebody's cell and just cut DNA randomly. It was going to go into a cell and it was going to be able to cut DNA at exactly the spot that scientists wanted it to. And that was what's important because it allowed them to regulate specific genes, not just, you know, random ones. Once you cut the DNA, though, then you have some options. So with that cut DNA, that's going to turn the gene off. It's you know, you can have a polymerase enzyme that might still start at the primer, but it's going to get to that point where the DNA isn't continuous and it's just going to stop, and your DNA, your protein synthesis would stop there. So that's a way to turn a gene off if you're synthesizing a protein that you don't need to be making. It also allows you to add new nucleotides in there to correct a gene or insert a new gene if that's what's needed. People with PKU, they're not able to make certain proteins, and so it allows them to kind of make that protein. So lots of different examples for this. They've been able to take the malaria gene out of mosquitoes. They can manipulate mosquito genomes so that way they no longer have that carrying capacity. You can alter plants to boost their yield, improve disease resistance, and survive drought. You probably heard about GMO organisms, genetically modified organisms. This is one of the ways that they do that, is they go in there with the Cas9. They can modify those gamete cells and insert those herbicide genes or drought-resistant genes, make strawberries redder and sweeter, those kind of things. All kinds of things. You can keep kind of reading through these. It's very interesting. The applications are incredibly interesting. But again, just kind of keeping in the back of your mind. All right, well, How are these things passed along? How are these things used? Who has access to these kind of things? What are the long-term outcomes of some of this stuff? It's just always good to have in the back of your mind. Science sometimes can be singularly focused on progress and not really think about what it's actually doing in the moment. And without context, science isn't really helpful. So with these Cas9 enzymes, again, CRISPR enzymes are able to start modifying our genome, start modifying things, but we can kind of still use those same principles. This idea of finding guide RNA, finding specific sequences in specific regions of DNA. We use that for DNA profiling. So we can use DNA profiling for genetic differences. What we're seeing here are SNP, single nucleotide polymorphisms, similar to the example that I was using, like with people that have diabetes. Once you know that someone has diabetes, you can then look in their genome, figure out what common sequence they have with that other people with diabetes have, and say, okay, well, there's your insulin gene right there. Similar to that, places like, or the genetic testing companies like 23andMe and Ancestry.com, all those kind of things, where you send in a little saliva sample. What they do is that they use self-reporting. So people that have lived in Ireland for 10 generations, and they have proof that they have been in Ireland, their family has been in Ireland, they are as Irish can be. 23andMe will go and they will get their DNA. They'll collect their DNA. And then they will look at all of the people that say the same thing. All these people that say, oh, yes, I'm from Ireland, I'm as Irish as you can get. Well, we look at all their DNA, we see what commonalities do they have? What SNPs, what single nucleotide polymorphisms in these, 15 different, 20 different, 30 different genes that we're looking at, however it may be. How many times do people that say they're from Ireland have the same change, the same polymorphism, the same nucleotide, specific nucleotide in that gene at that same place? Because those individual little ones are going to be different with people from Ireland that are very, very Irish versus people that are from Bulgaria or Hungary or Turkey or Chad or Botswana, right? All of those places, they're going to have the same genes, but in certain parts of those gene sequences, there's going to be individual nucleotides that indicate what region you're from. And so by taking self-reporting from people in those countries, they can then compare someone like myself, that's only been in America for a couple of generations. My family is kind of scattered from all over, like everyone, right? They can take our DNA and they can start to say, okay, well, how many of these little changes do you have? Okay, well, you have 4 changes that relate to Ireland, and then you have 7 that relate to France, and 4 that relate to this place, and blah 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 blah, all those different things. And that's how they start to say, okay, well, you're 20% Irish and 30% African and 26% Hispanic, and all those kind of different things, right? The combination that you get. They're looking at these single nucleotide polymorphisms, these individual little changes that are consistent across populations, especially as you're only looking at one nucleotide. It's not very likely that only one person would have one nucleotide different. What is very likely are STRs, short tandem repeats. So think about those names. Single nucleotide polymorphism. A single nucleotide can be changed, and that change will be consistent across populations. STRs, short tandem repeats, are actual sequences of DNA, sequences of nucleotides that repeat themselves. And this isn't necessarily related to a specific gene. This is just a part of your DNA where your body, again, only 1.5% of our DNA is coding. So we have regions where it's just where it's just iterative, nucleotides kind of strung together. And so it's really not likely for me and you and you and you and the rest of the world to have really matching STRs. We might have some that match because, we're talking about 8 billion base pairs. But it's really not likely that we would have multiple matching STRs. So STRs are used for crime scene investigations. These are used for genetic testing in that kind of sense to determine if someone is guilty of a genetic crime or a crime based off of their DNA. Because it's really unlikely to have multiple STRs from a suspect or from a crime scene and then matching your suspect. If the STRs match, that DNA was there. That's your DNA. Doesn't mean you were there. Doesn't mean you committed the crime, but it means that your DNA was there. So that's always specifics. And also, one of those things, people put a lot of weight into DNA evidence in crime scene investigations. We've gotten really good. We've gotten a lot better at our DNA to the point that we've been able to prove that some of the original convictions that people got off of DNA were incorrect. Because especially with a lot of the sequencing, it was happening to be done by hand, by people going through, and people make mistakes. Computers make mistakes too, but people make a lot more mistakes sometimes, especially with this kind of redundant work. And so, DNA is one of those things where it was really, really held as a standard, and then it's kind of had its kind of skepticisms. And so it's always interesting to see how DNA plays a role in those investigations. Okay, moving on. Stem cells. So talking about stem cells real quick. So this is one of those, again, that if you know a lot about, if you've kind of heard about many controversies with DNA technology, stem cells are definitely up there. This is another one of those that, when I was growing up, we were just starting to work with stem cells and manipulate stem cells. And so there's a lot of controversy, especially around embryonic stem cells and people not quite getting the difference between embryonic and adult stem cells, and also how embryonic stem cells were being harvested, or that's always a rough word to use, being collected. So just kind of keeping that in mind, if you are interested in that, please go read actual scientific articles, research about that. Please read verifiable news resources if you're looking through the news media, because it is a very sensitive topic, and so people can be really polarized and opinionated and kind of lose some of the actual facts of it. So start with the facts and then start to see where your opinion goes from there. So what are stem cells, though? That's the important thing to think about. Stem cells are undifferentiated cells. And so with an embryo, so what is an embryo? How about that one? There's a question for you. What's an embryo? After the sperm and egg come together, they make a single cell. That's called a zygote. That zygote then will go through about a week or so of changes, of growing, of dividing, until it implants in the uterus. After about 10 days, 2 weeks or so after fertilization, it's going to be in its embryonic period. And so an embryo refers to a fertilized egg about 3 weeks after fertilization. The embryonic period is week 3 to week 8 after fertilization. And during the embryonic period, you have a lot of large structures kind of being formed, the rudimentary structures of a fetus being formed. So you have your pre-embryonic period, which is fertilization, the first about 2 weeks after fertilization. The embryonic period is weeks 3 to 8, and then the fetal period is weeks 9 until birth. And so the embryonic period is really the time where you have the basic systems being kind of established. And then during the fetal period, that's when a lot of them start to grow and finish kind of, or start to polish off some of their development. So an embryonic cell, an organism that is in its embryonic stage, has stem cells that are going to become everything. The cells that start off, you start off as one single cell, and that single cell becomes the trillions of cells that you have. And it's because your stem cells have this ability to divide but stay alive. So typically with a cell, when we talk about mitosis or meiosis, that parent cell is going to divide, and one cell becomes two cells. So technically that parent cell is now kind of split between two. But with stem cells, what happens is that one cell, it will divide into two cells, but one of those cells will stay alive, will actually not be functional, and it will just keep dividing. And so it will kind of keep producing one cell, and that second cell is your stem cell that stays alive. And it just keeps producing more and more of those same kind of things. Stem cells, if they are, again, depending on the type of cell they can become anything, or they can become several things. Embryonic stem cells are totipotent. One of the reasons why they're so helpful in research is because embryonic stem cells can become any kind of body cell, depending on the chemical stimulus that you give it. There are certain chemicals that will bind to receptors on cells, and that will start to differentiate their development if they want to become a kidney cell or a liver cell or a pancreatic cell or whatever it may be. A neural cell, it's all based on kind of the chemical signals that they're getting. And so you can take an embryonic cell, an embryonic stem cell that hasn't differentiated, give it certain signals, it will grow into something, and then you could potentially put that into a person, actually use that as a treatment for some kind of condition or disease. Because they're going to give rise to all body cell types, neurons, muscle cells, red blood cells. So neurons especially are very slow to divide. Some of your neurons will never divide. Once they've been created, they were created during your embryonic period, and if they get damaged, they stay damaged. Your body might make new ones, but they don't really repair some of them. Not the case for all neurons. Some neurons are going to repair, but it's very slow. It takes years for the neurons to really repair. Well, embryonic stem cells, you could induce their development into a neuron, insert that into somebody, and then really speed up that development. So it has these potentials for treatment. Again, the controversy used to come from the fact that for things like IVF, when people, because of the early stages of IVF, it was very unpredictable, and so they would fertilize 6 to 8 embryos, implant all of those, and only a couple would actually go towards viability. And so some of them, they would actually take those cells because they knew they weren't going to be viable as an embryo, as a born fetus, and they would then take those cells and then use those for research. So there were some controversies in there in how they were harvested, but they weren't actually grown towards maturity. They were not a full-grown organism. They were still very much in that pre-embryonic stage, where again most people would not even know they're pregnant. So it's again, look at the facts, look at the research, and allow that to start to guide your opinions is my recommendation. What people are much more comfortable with are adult stem cells. Adult stem cells are what we call pluripotent. So while they can't become everything, they can become a lot of different things. An example of that is your bone marrow cells. You have cells in your red bone marrow that are pluripotent. They're going to become your white blood cells, all your different leukocytes, your fat cells, and your red blood cells. So they can divide into a bunch of these different things. So you can use adult stem cells. You can, again, send them different chemical signals. It will induce them to develop into a specific type of cell. And let's say you have somebody that has some kind of immune condition where they aren't producing white blood cells. Oh, well, we can use this therapy and we can start to induce, put these adult stem cells into your body, and they will reproduce themselves as white blood cells. All we have to do is just send that signal and they'll start to grow like that. So it's a way to kind of supplement that. And again, because they're from adults, you can have full consent for harvesting. It takes, it takes that kind of out of it. One other thing, cloning, again, another kind of very controversial variant. This chapter is always kind of fun because it's always like, it brings up so many different little things. And there's, again, popular media resources, scientific resources, and then you kind of have to figure out, all right, where do you feel comfortable in that once you're an educated individual in these topics. So cloning is an interesting one because we're creating exact copies of an organism. I'm trying to think about who it was. There was just an actor who was talking about, maybe it was talking about how they've had their dog. Oh, it was the football player Tom Brady. Yeah, Tom Brady was talking about how he's had his dog cloned like so many different times. Barbara Streisand is famous for that. She's had her dog cloned so many different times. You're not going to have the same dog. That's the thing. It can be the same genes, but it's not raised in the same environment. It's not going to be the exact same conditions, and so those things really do influence the expression of DNA. If you remember that epigenetics conversation from chapter 10, your environment really expresses your phenotype, really changes your phenotype, and so it's interesting science. How about that? So with clones, what you see here are asexual clones. So essentially all you do is take the full genome from one organism, put it into an unfertilized egg cell, and then put that egg cell into, implant it into a uterus to allow it to grow and develop. In this case, we're using plants. This picture is plants, so there's obviously no uterus. The uterus is the agar. It is the solution in which has all the nutrients that then allow that one little cell to grow into these plants. As it says, plants are very easy to clone. They kind of are made for that. That's one of their great things. Plants are readily clonable. Animals, not so much. Animals don't like to be cloned. It's much more difficult. You have to, again, it's a skill, being able to clone organisms. You have to figure out just the right conditions because you're working with these animals whose bodies are going to detect if something is starting to get out of homeostasis. So down to, with an animal that's going to be like the surrogate, the animal that you actually implant the cloned, the cell, the embryo that you want to, that animal, it has to be primed for pregnancy. You can't just take a surrogate mother who is not ready for implantation and put something into her. There has to be an entire fertility treatment beforehand to kind of line up those cycles so that the surrogate mother and the fertilized egg are both ready at the same time. Usually when we're talking about in vivo, meaning when man and woman are doing it together, creating reproduction together, It's about 24 hours after ovulation. So after the female is ovulated, there's about 24 hours where she's really optimal for fertilization, where fertilization is most optimal to happen. Because fertilization has to happen up in the fallopian tubes. It doesn't happen in the uterus. And so sperm has to go up through the uterus, up the fallopian tube, and then meet the egg there. And so there's a very kind of narrow window of about 24 to 48 hours of the optimal time. Because if the egg gets too far down the fallopian tube, or if it's in the uterus by the time it meets the sperm, that's too late. Fertilization is too late. The chemical signals will be off, and you're going to menstruate, and you're going to wash everything out, including that fertilized embryo. That might have been viable. But if the timing is off, then it's not going to work. So it's very, very subtle signal. So animal cloning is very difficult. The first cloned sheep was Dolly. The first cloned animal was Dolly. Again, I remember this. It's always weird getting to teach about it now. Because I was learning about it in school as was happening. So what happened is they took the animal to be cloned. They had their organism they wanted to clone, and because that animal was fully formed, they didn't take a gamete. They didn't take their sperm or egg cells. They took just one of their somatic cells. They took one of their regular body cells that had a full complement, because every cell has a full complement of your genome, except for your gametes. So they took one of its somatic cells and they extracted the nucleus from that, because that's where the DNA is. So they just pop the nucleus right out of one of those cells, right? They did this with multiple cells. We've learned this. They don't just do it once, they do it a bunch of times. So they took the nuclei out, and then they took a donor egg cell from another animal. So you start already with two different animals. You have the animal to be cloned, and then the donor animal. Well, they take the egg from that donor animal, remove any genetic material, because it is a gamete, so it's a haploid cell, it's half the DNA. They toss that out of there, and then they put your donor DNA into your donor nucleus into your egg. So now you skip fertilization. That kind of was your fertilization process, putting the donor nucleus into the host egg, or into that egg kind of. It's essentially, can we say eggshell? Is that not trying to be ironic? But into the casing of that ovum, and now you have a fertilized egg. Essentially, you have an organism that is ready to grow. You have a single-celled organism, because for an organism to grow, as we said in chapter 10, you need a diploid cell. A haploid cell, if an organism, if human only has one set of chromosomes, we cannot grow. We're not viable. We need sperm and egg to come together. We need those two haploid cells to make one diploid cell. That cell can then start to make proteins, divide, and grow. So when you take a nucleus, a somatic nucleus from a fully grown adult animal, and put that into an ovum, you've now just recreated, you essentially just put it back to its zygote stage. You kind of rewound it to its zygote stage, and you have a single organism that can then go through all those stages of development, all of those divisions. What's again tricky is that there is a specific stage of this pre-embryo that is optimal for implanting in the uterus. So again, this timing has to be right. Scientists will observe this embryo after they've kind of created the zygote again, the single-celled organism. I don't know if you can call it a zygote since it's cloned. That's an interesting question. But once they take the single cell and it starts to divide, it has to get to what's called the blastocyst stage. It has to go through about 8 different divisions. And at that point, because that will mimic the time that it would normally take from a zygote to go down the fallopian tube, it takes, again, about 7 to 10 days. So that 7 to 10 days, it's going through all those different divisions. It gets to its blastocyst stage. That's the stage you need this clone cell to be at to implant it into your surrogate, while at the same time making sure your surrogate's uterus is at the appropriate stage for implantation to happen, and it's not going to reject this donor egg. Very complicated stuff that has to happen. All right, take just a moment to that. I'm talking fast, I know, but it's exciting stuff. If everything works, if that embryo implants into the surrogate's uterus, the uterus accepts it, and it can grow, and it grows full to term, and there's no issues, then you can start to create, that embryo can develop into a lamb. A clone of the one that was original. Dolly, the original sheep, I don't believe that it lasted for very long. I think it lasted for a few years, and then it had maybe some health conditions that were underlying, I think, in the original host, but which came about in the clone because, again, epigenetics. You're raising them in different conditions. Now we've gotten much better at cloning. Again, they do it to people's dogs, celebrities' dogs all the time. Humans have not been cloned yet, to my knowledge, but the world's a big place, and labs can be small and tiny. So it's kind of one of those kind of scary things to think about. This technology is out there. People have it. They have it out there. And hopefully people are being ethical with it. So one other aspect is genetic testing. This is another one of those that at first it seemed kind of nice and harmless, and then you kind of think about some of the other implications for it, and again, it's the idea that, okay, let's hope that people are using this responsibly. So genetic testing can be used to detect diseases, similar to kind of gene sequencing. You take like the idea of gene sequencing and then essentially just make it a probe. Now you just make it something that you can look for. So if two individuals, this is not relegated to IVF at all. This is just two people that are about to have a child can go to a fertility specialist. They can have genetic testing done, and they can kind of like we did with chapter 10 or the last lab, the human genetics lab, I mean, they won't do Punnett squares, I'm sure, but they can analyze the two individuals, the two parents' DNA, and they can say, okay, these are the chances that your children might have these conditions. These are the ones you need to look out for. These are the things that, might you want to be aware of. Typically, that's for diseases. It's for diseases that would be called non-viability in the embryo. Rarely, and really we can't, it's very, very difficult to try and predict for eye color, hair color, height, intelligence. Those things really can't be predicted because they are so influenced by environment. But with a DNA probe, they could do things like breast cancer detection. If you are curious, if you have a history of breast cancer in your family, you might be interested to see if you have the gene for that. Again, they kind of took the people that were diagnosed with breast cancer, they looked at their DNA, they said, oh, well, there's the gene for it. And so now we can test for that breast cancer gene, that particular type of breast cancer. Always important to highlight that. But they can look and they can see, oh yeah, that's what it is. They can create what's called a probe, a complementary section of DNA that will bind to any DNA that has that matching gene. This one is looking at cystic fibrosis. Cystic fibrosis is a condition that causes mucus to accumulate in the lungs. They don't have a correct protein pump, and so they're mucus will accumulate, and so it can be a fatal condition. And so with this, they can test someone for cystic fibrosis, and if they have that, they can then start to treat them for that. They can do this pre-implantation genetic diagnosis, the PGD, is again a way that you can take an embryo, pull out one of its cells, look at the DNA, and start to determine if this is going to be a viable embryo. If it is going to be viable, is it going to have any kind of conditions? And some genetic conditions, like we saw in the human genetics lab, some do show what we consider kind of an incomplete dominance or a codominance, and they can have varying degrees of intensity. And so PGD can start to determine that. So for something like cystic fibrosis, they do have developed a gene therapy for it. It's an inhaler because cystic fibrosis really affects the lungs. It affects the rest of the parts of the body, but the real pathological conditions show up in the lungs. And so what happens is someone can hit their inhaler, and when they inhale it, they're actually taking in these viral particles that have the gene they need. And because viruses are going to infect our cells, in this case, they're infecting this patient, with the gene they need to make the proper protein. So this is going to then get into their somatic cells. It's going to change the DNA in their somatic cells and allow them to express the correct protein. The issue here is that some of those cells may be able to then pass on that modified DNA to their own daughter cells as they go through those mitotic divisions. They could potentially pass that on, or you might have to re-up the therapy. I'll be very honest, I'm not quite positive on the cystic fibrosis gene therapy. If it's like an initial, treatment every 2 weeks for 3 months, and that's enough to give you kind of a high dosage, or if it's a once a year kind of reinoculation, I'm really not sure. That would be something maybe with my holiday break, I'll go and look that up. But in this case, this is one of those things that I want to highlight, that we're modifying somatic cells. Those aren't gametes. If you're not modifying someone's gametes, if we were to go in and modify the sperm and egg stem cells that then create all of your sperm and ovum, if we modified those stem cells, they would then pass on that modified gene. But without modifying sperm and egg, you're not going to be passing that kind of thing along, most likely. Right? There's if someone develops a new technique, maybe that technique then spreads to the sperm and testicles, to the testicles and the ovum, and kind of creates some kind of modified gametes. But again, that's why we kind of always go slow with these DNA technology treatments. All right, thank you so much.